Hello everyone, welcome to our video. Have you ever wondered how genetic information flows from DNA to protein and how recombination contributes to genetic variation? So in today's video, we are going to talk about DNA and gene expression.
Hello everyone, my name is STB1033, my metric number is 112592. Today I will be presenting on DNA and RNA structure. DNA, also known as deoxyribonucleic acid. It contains deoxyribose sugar, phosphate and a base. It is a long molecule made up of monomers called nucleotides. In DNA we have bases such as adenine, guanine, cytosine and thymine. DNA is a double helix and it has two strands that twist around each other. Each strand of DNA can be millions of base pairs in length and is coiled up to make chromosomes. Now, we will look into the structure and bonds in DNA. The phosphate end is called the 5' end because the fifth carbon is closest to the phosphate group, whereas the sugar end is called the 3' end because the third carbon is closest to the sugar. The strand is held together by hydrogen bonding between the bases. A purine, adenine and guanine must base pair with pyrimidine, cytosine and thymine. A phosphodiester bond is a strong covalent bond that joins nucleotides together in DNA and also in RNA. It forms the sugar-phosphate backbone of nucleic acids. As we can see in the picture given, the adenine and thymine base pair have 2 hydrogen bonding, while the cytosine and guanine base pair have 3 hydrogen bonding, and the strands are in antiparallel to each other. The function of phosphodiester bonds is to hold nucleotides together and also to provide structural stability. Now, we will move on to RNA. RNA, also known as ribonucleic acid. Ribonucleic acid consists of a phosphate group, pentose sugar, and nitrogenous bases. The pentose group in RNA is called as ribose. RNA monomers use ribose sugar. Alright, now let's move on to the bases in RNA. RNA consists of adenine, guanine, cytosine, and uracil. It is a single-stranded RNA, and also thymine in DNA is replaced by uracil in RNA. I hope you are clear with that. In this slide, we will look into the types of RNA. Firstly, we have messenger RNA. The mRNA is a transcript copy of a gene that encodes a specific polypeptide. Secondly, we have transfer RNA, tRNA, transfers amino acid during protein synthesis. Thirdly, we have ribosomal RNA, also known as rRNA, forms the structural and functional core of the ribosomes. And we also have a quick recap on RNA in the picture given in the slide. In this slide, to make it easier for someone to remember the base pairs, we have mnemonic for DNA and RNA. To remember the base pairs of adenine and thymine, we can remember it as apple on the tree. And as for the guanine and cytosine base pair, we can remember it as cars in the garage. And for the RNA, where the thymine is replaced by uracil, we can remember it as apples under. So, I hope this helps you.
My name is Changlao Yi, and now I will present on the genetic code. So what is genetic code? Genetic code refers to the sequence of nucleotides in DNA and RNA that determines the amino acid sequence of proteins. The code is read in groups of 3 nitrogenous bases known as triple code or codon. Since there are 4 different bases, combinations of 3 bases produce 64 possible codons which code for 20 amino acids. For example, the codon AUG codes for amino acid methionine. It is known as the start codon because it signals the beginning of protein synthesis. Among all 64 codons, 3 of them are stop codons. These do not code for amino acid, but instead, they signal the end of protein synthesis. The genetic code is stored in the order of bases along the DNA sequence within genes that contain the instructions needed to produce specific proteins. Another important feature of genetic code is called degeneracy. Degeneracy means that one amino acid can be coded by more than one codon. For example, amino acid proline can be coded by CCU, CCC, CCA, and also CCG. However, Although one amino acid may have multiple codons, a single codon will only code for one specific amino acid. For example, CCU always codes for proline, while AUG always codes for methionine. This property is known as the non-ambiguous nature of genetic code.
DNA carries inherited information and must be able to copy itself exactly to ensure that the genetic details are passed down accurately to the next generation of cell. So, every single second, your body is copying your entire genetic blueprint. This is DNA replication, and the entire process is driven by 3 fundamental steps: unzip, copy, and glue. Step 1. Unzip. First, helicase act like a molecular zipper, slicing through the light blue double helix to separate the DNA strands and create a replication fork. Step 2. Copy with direction. Next, DNA polymerase move in to build a new strand, which is in a darker blue color. But it has a strict rule. It can only synthesize DNA in the 5' to 3' direction. This is because the original template strand run in opposite direction, which is 3' to 5'. One new strand is made continuously. The other, however, must be built backward in short, choppy segments known as Okazaki fragments. Step 3. Glue. Finally, ligase act as a molecular glue, stitching those Okazaki fragments together. And the brilliant result? 2 identical DNA double helix, following the semi-conservative model, are formed. Each new molecule contains 1 original parent strand and 1 brand new daughter strand. And that's replication of DNA, precise, directional, and essential for life.
I'm Evangeline, and I will explain about the transcription. The definition of the transcription is a process by which genetic information on DNA is copied to form mRNA. It occurs at nucleus, and it is divided into 3 stages. The first stage is initiation, the second stage is elongation, and the last stage is termination. For the stage 1 initiation, RNA polymerase binds directly to the promoter. The enzyme unwinds the DNA double helix, and the RNA polymerase also initiates mRNA synthesis at the start point using the 3' to 5' DNA template strand. Stage 2 elongation. To build the strand, RNA polymerase moves along the template strand, adding RNA nucleotides to the 3' end of the growing chain. Synthesis occurs strictly in the 5' to 3' direction using complementary base pairing. Behind the enzyme, the DNA double helix rewinds and the newly made RNA peels away. Stage 3 termination. To release the transcript, RNA polymerase transcribes a specific polyadenylation signal sequence AAU-AAA in the RNA transcript. A specific nuclear protein binds to this sequence and cuts the transcript, releasing the pre-mRNA. RNA polymerase detaches from the DNA template, completing the process.
At the ribosome, translation occurs. The ribosome reads the codons on the mRNA. Transfer RNA, which is the tRNA, brings specific amino acids to match each codon. The amino acids join together by peptide bond to form a growing polypeptide chain. When translation is complete, the polypeptide chain is released from the ribosome. This allows cells to produce a protein needed for life.
Hello everyone! Now, we will move on to crossing over. Crossing over or recombination is the exchange of DNA segments between homologous chromosomes during prophase 1 of meiosis. This process creates a new combination of genes, genetic variation in offspring, and also differences between individuals. So, what are exactly homologous chromosomes mean? Homologous chromosomes are pairs of matching chromosomes in a diploid organism, where one chromosome comes from the mother and another chromosome comes from the father. They carry the same genes but may contain different allele. For example, one chromosome may carry brown eye allele and another may carry blue eye allele. I hope you're clear with that. Now, exactly what crossing over contributes to? Firstly, crossing over contributes to different eye color, hair type, and height variation, also facial differences. This is why even siblings look different because of the recombination. In previous slide, we know that crossing over is an exchange of portions of chromatids between homologous chromosomes. But how does it occur? In here, we will see on steps of crossing over. First, in prophase 1 of meiotic cell division, homologous chromosomes pair up, as we can see in picture 3. Then, non-sister chromatids may wrap around each other, forming a cross-shaped structure called a chiasma. At the chiasma, the chromatids break, exchange, and rejoins. This is what we are looking in picture 4, 5, 6, and 7. Once they rejoin, crossing over is completed.
That's all for today. Thank you so much for watching our video.